Hi everyone, welcome. Today I will be presenting our paper, Mapper Interactive, a scalable, extendable, and interactive toolbox for the visual exploration of high-dimensional data. Here's an overview of the talk. I will start with the motivations and contributions of our paper, and then review some of the background concepts for our work, specifically, the construction of mapper graphs. After that, I will go through the design details, and demonstrate 3 different use cases to show the utility of our tool, and finally talk about the conclusion and discussion. This project was born out of a need for a framework for interactive analysis and visualization of high-dimensional point cloud data, using the Mapper algorithm, a popular topological data analysis tool. Our framework makes the Mapper algorithm accessible to non-specialists, and those with a passing knowledge of programming concepts and TDA. It also gives specialists the ability to extend the system, by adding analysis and visualization components in a modular fashion. The toolbox comes with both a web-based interface for online computation, and a command line API for offline computation of mapper graphs. Our major contributions consist of 3 aspects: the extendability, interactivity, and scalability. For the extendability, we provide 2 modes for different user groups to easily extend the framework. For interactivity, we present an interactive visualization system for on-the-fly computation and exploration of mapper graphs across a range of parameters. For scalability, we present a simple but effective strategy for speeding up mapper graph computations. In particular, the command line API can compute mapper graphs for 1 million points of 256 dimensions in about 3 minutes, which is 4 times faster than the vanilla implementation. For a better understanding of our framework, I'm going to review the mapper construction algorithm, which is first introduced by Singh et al. The mapper algorithm is a popular tool from TDA for extracting topological summaries of high-dimensional point clouds. It is rooted in the idea of partial clustering of the data, guided by a set of functions defined on the data. Given a high-dimensional point cloud, X, a cover of it is defined as a set of open sets, such that X is a strict subset of the union of the open sets. The one-dimensional nerve is a graph, where each node represents a cover element, and there is an edge between 2 nodes if their intersection is not empty. This figure gives an example, in which X is a 2D point cloud sampled from the silhouette of a snowman. The figure shows the cover of X in A, which consists of a collection of rectangles. And the corresponding one-dimensional nerve is shown in figure C. For example, the cover element U1 on the left becomes the node 1 in the nerve, and U2 becomes node 2. Since the 2 rectangles intersect with each other, there is an edge between node 1 and node 2 in the graph. In the classic mapper construction, a set of scalar functions, which are called filter functions, is used to guide obtaining a cover. Given a cover V of the scalar function, we can obtain a cover U of the point cloud, by considering the clusters induced by points that have function values belonging to each cover element Vi. Almost any clustering algorithm can be used to obtain the clusters. In this paper, we chose to use dbScan, which is a density-based clustering algorithm, and eliminates the need for specifying the number of clusters. The 1D nerve of cover U is then the mapper graph of X given F. Now let's go back to this figure. We can see that in figure B, the point cloud is equipped with a height function, and the cover elements from V1 to V6 form the cover of this height function. To find the cover of the point cloud, we first need to identify all points belonging to each Vi, and then we apply the clustering approach to obtain clusters. Such clusters then form the rectangles in A, which are the cover elements of the point cloud. And the 1D nerve shown in C is then the mapper graph of the point cloud. This figure shows the user interface of our visual system. A is the graph visualization panel, which visualizes the resulting mapper graph of the input data. It enables basic interactive operations such as zooming, dragging, and panning. The current mapper graph is computed from the snowman point cloud shown in D. B is the selection panel, which enables users to select a subset of mapper graph nodes, under 3 different selection modes. The selected subset of nodes can then be used for further analysis, such as linear regression and dimensionality reduction. C is the control panel, which provides parameter controls for computing mapper graphs on the fly. It also includes several data wrangling options, such as normalizing the input data and choosing a subset of columns to form the point cloud. There are 3 data selection modes. A shows that under the Select Nodes mode, users can select any number of nodes in the mapper graph. B shows that under the Select Clusters mode, users can select connected components of the mapper graph. C shows that under the Select Paths mode, users can specify the start and end point of a path in the mapper graph, and select the shortest path between them. And D shows that this path can be extended by selecting another endpoint. In terms of extendability, we provide 2 modes for different user groups to easily extend the framework. Under the Novice User mode, all the users need to do is to describe the new module information within a template file, and the system will detect and generate all the new modules automatically. Currently, the system allows the addition of supervised and unsupervised learning algorithms that are available in scikit-learn. Under the Expert User mode, users with programming experience can modify the template function to construct multi-step analysis pipelines. The figure shows adding a TISNI module to the interface. A is the information to be added to the template file under the Novice User mode, and the added module is shown in B. C shows that under the Expert mode, we can further customize the module by coloring the points using the k-means clustering result. Our system is equipped with scalable backend computation, which speeds up the mapper algorithm framework. The backend mapper implementation is built upon Kepler Mapper. Kepler Mapper is a user-friendly implementation of the mapper algorithm. However, when the size of the point cloud is large, its default mapper graph computation does not scale well. The computational bottleneck happens during the dbScan clustering stage, in which the algorithm queries all pairwise distances. To speed up the implementation, the first idea is parallelizing individual clustering instances, that is, computing the clusters for the inverse map of each interval. We then push the parallelization even further, by pre-computing the distance matrix of points within each interval. Such a strategy is applicable to any mapper framework employing dbScan as a clustering subroutine. In experiments, we use 3 different datasets to perform runtime analysis for Mapper Interactive, against 2 of the state-of-the-art tools, Giotto TDA and Kepler Mapper. The ImageNet and Cipher datasets are created by passing input images to neural networks and collecting the activation vectors. The ImageNet dataset has 512 dimensions and 300k points, and the Cipher dataset has 256 dimensions and 3 million points. For the random vector datasets, we sample 10 million points of 128 dimensions. Each dimension is drawn from a uniform distribution. To demonstrate that our strategy will speed up any dbScan-based mapper framework, we also include a modified version of Giotto TDA and Kepler Mapper, which are speeded up using our strategy. We first show a comparison of peak memory usage. The GT star and KM star are Giotto TDA and Kepler Mapper with our speed-up strategy. In general, there is a space-time trade-off, as we pre-computed the distance matrices for faster computation. For all 3 datasets, Mapper Interactive has up to a 1.7 times increase in peak memory usage, compared to its vanilla implementation. Also, using our strategy, Giotto TDA has up to 4 times memory increase, and Kepler Mapper has up to 1.9 times memory increase. These plots show the runtime comparison. Each column is a different dataset. The first row shows the CPU runtime of the 3 different large point cloud datasets. The blue line shows the runtime of our framework. The orange and green lines are the original implementation of Giotto TDA and Kepler Mapper, and the red and purple lines are the speed-up versions of them respectively. It shows that both Giotto TDA and Kepler Mapper can be improved to achieve comparable performance with Mapper Interactive. In particular, for the ImageNet dataset of 300k points, Mapper Interactive achieves an approximately 6 times speedup against its vanilla implementation. With our strategy, Giotto TDA gets 9 times speedup, and Kepler Mapper achieves 7 times speedup. The second row shows the GPU runtime in comparison with CPU runtime for Mapper Interactive. The blue line is CPU runtime and the orange line is GPU runtime. There is a roughly 2 times speedup of CPU implementation, in comparison with the CPU implementation. We demonstrate the utility of Mapper Interactive through 3 use cases. Our first use case is to analyze and compare COVID-19 trends in the United States. The dataset contains the daily records of COVID-19 cases in all 50 states, from April 12 to September 18 in 2020. It contains 9,240 data points. Each point corresponds to a daily record for a given state. For each state, it contains 7 statistical measures, such as the number of confirmed cases, death cases, and active cases, etc. We first compute an initial mapper graph using all data points. We use all 7 columns to form the point cloud, and use the number of recorded days as the filter function. The size of each node is encoded by the average number of recorded days. And in Figure B, the nodes are colored by the number of confirmed cases. The mapper graph indicates that certain states are shown to be separated from other states, and form their own connected components, such as New York and Massachusetts, which implies that their epidemic trends may be quite different from others. To further investigate why and how these states are separated from one another, we select 9 representative states with the largest number of confirmed cases, and compute a second mapper graph. The second mapper graph shows that all the states become separated from each other after certain branching points. By comparing the line graph on the right, we can see that the order by which each state is separated is related to how different its curve is from other states. For example, as shown in the line graph, the curve of New York deviated the most from other states, so in the mapper graph, it is not connected with any other state at all. New Jersey shows the second highest deviation besides New York in the line graph, so it splits from the main branch at node 4 in the mapper graph. Arizona and Georgia, as well as Florida and Texas, are 2 pairs with similar trends in the line graph, so their nodes are the last ones to be separated from the main branch. Through the mapper graph, we can gain insights of the epidemic trends in different states in a fast way. We can also discover when their trends start to diverge, by looking at the nodes at the branching points. Our second use case is to visualize neuron activations collected from a categorical dataset. The data is collected by passing input images to a neural network, and collecting activation vectors from the last layer of the network. We then treat these activation vectors as the inputting high-dimensional point cloud to compute the mapper graph. There are 50K images from 10 image classes, including ship, truck, automobile, etc. Each image corresponds to an activation vector with 512 dimensions. The resulting mapper graph highlights the separation of image classes. The pie chart on top of each node shows the proportions of categories within each node. As we can see, all the classes are separated from each other after some branching points. This mapper graph also highlights the relationship among the different classes. For example, the deer and horse images first emerge from the branching node 18. Then, the 2 classes are separated into 2 branches at node 32. The branching order indicates that the deer and horse images are more similar than images from other class categories. We then perform additional analysis to further advance our understanding of the dataset. First, we added a PCA analysis module to compare the PCA result with the mapper graph result, as they both perform dimensionality reduction of the high-dimensional data. Compared to the mapper graph, the PCA projection does not separate the classes well, and the relationship between different classes is not well depicted. In addition, for such a large dataset, parameter tuning can be difficult and time-consuming. We then added another new module for tuning the epsilon parameter in dbScan clustering. We plot the sorted distance distribution of the 5th approximated nearest neighbors of each data point, to figure out the most appropriate epsilon value. The right epsilon value should maximize the number of points having at least 5 neighbors, while making sure the distance doesn't go too large. In this case, we can see an appropriate epsilon value should be around 8. Our third use case is to provide alternative ways to explore the results from a breast cancer study. We use the two breast cancer datasets, the NKI dataset and the GSE dataset. The NKI data contains information from 272 breast cancer patients. For each patient, there are two types of variables. The first is gene expression levels, and the second consists of other medical records, or physiological measures. The GSE data consists of expression levels of over 20,000 genes from 286 patients. In the previous study, the MAPR graph is used to study the subgroups of breast cancer patients. We are interested in the expression level of one gene, called BSR1, which is usually positively correlated with the prognosis. Patients with high expression levels usually have a better prognosis, and are more likely to survive than patients with low levels. However, among all the patients with high levels there are still subgroups having poor clinical outcomes. Our goal is to identify such subgroups from patients using MAPR algorithm. The image shows the MAPR graphs of both NKI and GSE data. On the left side, the nodes in the NKI MAPR graph are colored with either survivor or not. The nodes in the GSE MAPR graph are colored with either the patients were relapsed or not. On the right side, the nodes are colored with the gene expression levels. The size also indicates the expression levels. It is interesting to observe that the two resulting MAPR graphs consist of similar structure for the survivor or non-relapsed patients. In each graph, the blue connected component on the bottom contains a branch of nodes, with low average expression levels, which defines a subgroup of survivor or non-relapsed patients. The result shows that we are able to visually identify similar subgroup structures, under two datasets generated from totally different experimental settings. Furthermore, we can easily extend the framework to perform new analyses of the breast cancer datasets. Since the NKI dataset contains medical records and physiological measures about the patients, we can make use of the regression analysis modules to explore interesting subgraphs of the MAPR graph. We first select the lower connected component, which contains clusters of the survivor, and apply the existing linear regression module to these clusters. The linear regression result indicates variables that have statistically significant effects on the expression levels, without affecting the patient's survival. We further explore which genes affect the survival of patients with low expression levels. We add a logistic regression to deal with the binary survival variable. We include the top 10 genes that are selected using a recursive feature elimination. The regression result indicates genes that significantly related to the survival of patients. Further analysis based on these regression results would be an interesting avenue to explore in a follow-up study. To summarize, in this paper we present MAPR Interactive, an interactive, extendable, and scalable toolbox for the visual exploration of high-dimensional data, using the MAPR graph. We provide both a web-based interface for online computation, and a command-line API for offline computation. Future work would include pushing the scalability boundary further, to improve the extensibility for both novice and expert users, and to explore automatic parameter selection strategies for the MAPR algorithm. The authors wish to thank Nathaniel Saul for comments on the initial prototype of our tool, Anantaraman Kalyanaraman, Mithun Kamarazaman, Balakrishnamurthy, and Umberto Lupo for valuable suggestions and discussions. Thank you for watching our presentation.